Hello everyone, it's my pleasure to present our group's new paper in American Chemical Society Omega, entitled Lysine Scanning of Arg10-Teixobactin, Deciphering the Role of Hydrophobic and Hydrophilic Residues. First, I'd like to give a brief introduction about Teixobactin. Teixobactin is a new antibiotic showing high activity against gram-positive pathogens. The first report about Teixobactin was in 2015 by Lewis Group, and they extracted this antibiotic from some gram-negative bacteria using a multi-channel device-eyed chips. This figure showed the formation of the bacterial cell wall, and from here we can see the mechanism of action of Teixobactin, where it inhibits the lipid 2, the precursor of peptidoglycan. Okay, let's take a look at the chemical structure of Teixobactin. As we see here, Teixobactin is a head-to-side chain cyclodipsipeptide. Why head-to-side chain cyclo? Because the cycle is between the alanine head amino acid and the L-androdosin side chain. And it is dipsipeptide because it contains one ester bond between the hydroxy side chain of D-threonine and the carboxylic group of isoleucine. Also, Teixobactin consists of 11 amino acids, 4 of them at the cycle and 7 at the tail. In the cycle, we have a non-proteinogenic L-androdosin, and in the tail, we have 4 L amino acids and 3 non-proteinogenic D amino acids, D-alloisoleucine, D-glutamine, and at the terminal, we have N-methyl D-phenylalanine. From this sense, the aim of this paper is studying the structure-activity relationship of Teixobactin using lysine scan as a new technique in the medicinal chemistry program. So, what is lysine scan? Lysine scanning of a peptide involves the preparation of a set of analogs where each one contains a lysine residue in different positions of the sequence while keeping the same stereochemistry. In this sense, 8 analogs containing L-lysine at positions 2, 3, 6, 7, 9, and 11 and D-lysines at positions 4 and 5 were prepared. And by performing the lysine scanning on Arg10-Teixobactin, we could identify the key amino acids in the peptide as well as we studied the importance of hydrophobicity of the molecule for antibacterial activity. In this slide, I'll discuss the synthetic strategy that I used to prepare those analogs. In the first step, after activation of CTC resin and loading the first amino acid, I used FMOC strategy to couple the following amino acids until get the tetramer peptide. Then, in the second step, the esterification step takes place between D-threonine and alloc-isoleucine. And after the ester bond formation, the alloc protecting group was removed using palladium chemistry, which is orthogonal with FMOC protecting group, to allow alloc-arginine PPF to couple. And again, by using FMOC strategy, I deprotect FMOC protection group and build up the tail until introducing BOC N-methyl phenylalanine. Then remove alloc from arginine and cleave from the resin in 1% TFA in DCM. This ratio of acid is essential to keep all amine protecting group for the N-terminal and side chain during the cyclization step, which takes place in solution for 24 hours. Then make the global T protection and purify to get the pure product to test MIC. As it showed from the MIC table, any substitution of the hydrophobic by isoleucine by the charged polar lysine led to total loss of the activity, while the substitution of the 3 polar uncharged amino acids, serine at position 3, glutamine at position 4, and the other serine at position 7, and also the neutral alanine at position 9, maintain the activity and in the case of lysine at position 3, improve the activity against B subclass. So from this report, we concluded that lysine scan as a new technique in medicinal chemistry is a good tool to identify the essential amino acids. And related to Teixobactin, keeping the balance between hydrophobic and hydrophilic amino acids is crucial for maintaining the activity, where substitution of hydrophobic amino acids led to total loss of activity, while substitution of hydrophilic amino acids retain or improve the activity. Thank you.